All right. In the interest of time, I'm going to skip most of my introduction. I work on Dialictus, which for this group is probably a group of bees that needs no introduction. As if you work on native bees at all, then you've seen this before. These bee bowls chock full of tiny metallic halictids. And if you're brave enough to try species identification, then you've seen this before, and you probably felt like this when you tried to key them out and found out how many species there are and how difficult they are to ID. So let's go ahead and skip to the interesting part. It all started with this bee right here. This is actually one of the most common species in the southwestern United States. If you do any collecting down there, it's really hard not to find it. They do the bee course, or they have done the bee course every year, and they always get a bunch of these down there. And it's also pretty easy to recognize for a Dialictus based on this kind of pale yellow tegula with this straight kind of hooked inner posterior margin. And that, along with the propodium having this oblique carina, puts it in what's called the Lasioglossum gemmatum species complex. This is a monophyletic group that's characterized by this tegula shape, and it also includes the more well-known tegulare group, which has this even bigger tegula with all these punctures on it. But here's the kicker. This really common, super easy bee is undescribed. It doesn't have a name, even though people are collecting it all the time. It's not that hard to recognize. And to put this in perspective, it's kind of like if this was undescribed and everyone just went around calling it bombus spo. So my PhD thesis is a taxonomic revision of the Dialictus of the Western Nearctic region. So I saw this bee and I thought, why not start with the revision of the gemmatum complex? Why not follow this bee down its burrow? Follow this dapper bee here down its Bee-Burrow and see where it goes. So historically, species identification in the gemmatum complex has been a fairly simple matter of geography combined with metasoma color. So if you split North America in half, anything in the east has historically been called Lasioglossum tegulare. Anything in the west has been tegulariforme, unless it has a red metasoma, in which case it was called Lasioglossum hunteri. This is a bit of an oversimplification. There are other species, there are other available names, but they're not commonly used. If you go into most museums, these are the three names you're going to see most frequently. Unfortunately, taxonomists are a bit like politicians because everybody likes us until we actually do something. So in 2009, Jason Gibbs split Lasioglossum tegulare, the eastern species, into 5. So it was actually 5 cryptic species. Then in 2018, Lasioglossum parvum, which is a Caribbean species in this group, turned out to actually be 4 species at least. So kind of have to wonder then, how many species are these Western ones really? And this is not a trivial question, considering that the southwestern US and northern Mexico is a bee biodiversity hotspot. It has the most bees, kind of basically in the world. And we know that the diversity is higher than what's been recorded there. There's 11 available names in the Western Nearctic in the gemmatum complex. And if you go on the Barcode of Life and you look up DNA barcodes, what you'll find is that there's 12, it puts them into 12 bins or operational taxonomic units. So it seems like there's higher diversity here, and it also seems pretty straightforward. 11 names, 12 bins. But spoiler alert, that hotspot right there might be a dumpster fire. So to answer this question and investigate this problem, I'm using what's called integrative taxonomy. which basically is a process where you generate hypotheses about species, and then you test them using at least two types of data. And the data types that I have available right here are morphology, so looking at specimens under the microscope, comparing morphological features, also generating cytochrome oxidase 1 DNA barcodes and uploading those on the Barcode of Life or BOLD website. And then I'm also looking at geographic distributions, which I use by georeferencing specimen records and then plotting them in Maxent, which gives you not just the known distribution, but also it predicts remaining suitable habitat where you don't actually have records. So what did I find out? If you define Lasioglossum tegulariforme as broadly as possible, that is any Western gemmatum group species that has a dark metasoma, it turns out that it's actually 14 species. 7 of those are already described, they already have names, and 7 of them are new. And it also turns out that it has this the real tegulariforme, the true thing, has this really easy diagnostic character. If you look at the first metasomal segment, it's got this distinct impunctate line down the middle of it, which is unique. So that was fun to discover because those sorts of easy characters are kind of rare in Dialictus. It also turns out that it has a much more restricted distribution than we've known before. So if you went on Discover Life right now and you looked up Lasioglossum tegulariforme, you would get a distribution that looks like this, but it actually looks more like this. It's only known from the Pacific Northwest and the California Central Valley and the Snake River Valley. And all those other records, it turns out, are a bunch of other species. Some of these are described, some of them are undescribed, but they're pretty much all geographically distinct. There's only one other species that occurs sympatrically with Lasioglossum tegulariforme, and that one is both morphologically and genetically distinct. So there's some pretty interesting allopatry going on here. So meanwhile, Lasioglossum hunteri, if you go on BOLD and you look up DNA barcode for this species, and you define it as broadly as possible, that is any Western species with the red mesoma, you find a pattern that looks like this. It comes out in 6 different places. So obviously, this can't be right. One of these is the real hunteri, and the other ones must be different species, unless there's something really crazy going on. So it turns out this is actually 7 species. 3 of them are described, and 4 of them are new. It also turns out that the real Lasioglossum hunteri is actually just the eastern species, Lasioglossum ellisii, it's a synonym. And at first glance, they look pretty different, but if I just flex my Photoshop skills for a bit here and take out all the reds in the image of hunteri, they don't look so different anymore. You can't really tell them apart. Furthermore, they have identical DNA barcodes. There's no way to separate them genetically. So what about geography? Well, I plotted several specimens, both dark-tailed and red-tailed. And there's a sort of general pattern where southwestern specimens tend to be red-tailed, but the boundaries are really fuzzy. And you also get a lot of intermediate cases like this, where it's partially red. So there doesn't really seem to be any clear separation here. And there's not really any evidence that can support a 2-species hypothesis. These other records, further west and further south, those are the new species. So, so far, this has all been pretty straightforward, but we're not to the bottom yet. The last frontier in the gemmatum complex is what I've called the Lasioglossum sticktaspis species complex, which is a subset of the gemmatum complex. They're very wildly morphologically variable. They can be dull or shiny, red or black, really enormous tegulae or smaller tegulae, and the geographic distribution looks like this. Now, this is probably multiple species on this map here, but I'm not sure how to separate them, so they're kind of all mashed together right now, but you can see they range all the way from Canada down into Central America. And of the available DNA barcodes, it looks like this. And if I try my best to separate them into different species, I get a pattern that looks like this, where these two orange and light blue species on the left here are fairly distinct. Those I'm planning to describe, but as for the rest of this mess, we're not in Wonderland anymore. This is like through the looking glass territory, and not even the classic one. This is like the 2016 movie remake. This doesn't make any sense. So with that, I need to break out the old cliche, more research is needed. And by more research, I'm thinking probably like alter conserved elements. But even without the Stichtaspis complex finished, and all these problems caused by rapid evolution and poor lineage sorting, I'm confident that there's 30 species in the gemmatum complex, 10 of those being new, and there's 19 total in my study region of the Western Nearctic. And I've also uncovered several interesting cases of allopatry with closely related species having distinct geographic distributions. So with that, just acknowledge my committee and my funding and all of the museum curators and people who were generous enough to lend these specimens.